Thank you so much for joining us today for the third annual is Knowledge is Pollinator Power webinar. And I'm very excited today to welcome folks from the mountains to the sea and all of our speakers joining us, and Becky Griffin down from Georgia, who is the project creator of the Great Southeast Pollinator Census. So we're going to learn about the census today, and we're going to learn some really cool pollinator knowledge that's been going around. So my name is Amanda Bratcher. I'm the horticulture agent in Lee County, which is just southwest of Raleigh, almost in the geographic center of North Carolina. We're 10 miles away from the geographic center. So when you're looking at the map, that's where we're at. This is actually a picture of all of the state coordinators except for the state coordinator for Alabama. We went on a knowledge sharing trip back in May to London, and Becky is such a great photographer. She made sure that we were well documented while we were there. So, and I'm very happy to have Dr. Charlotte Glenn, who is the state coordinator for the Extension Master Gardener program here in North Carolina. And she is, many of y'all may know her as we have a lot of Master Gardener volunteers here, but Charlotte is a phenomenal moderator. So she's going to make sure that if you have any questions, please throw them in the chat. Or actually, hey, that's not true. Look, I have a whole etiquette slide for this. So we are in a webinar. So this is a webinar format. So we cannot see any of you as folks who are joining us, but we can see your comments in the webinar chat, and we can see your questions when they come in the Q&A. So you should be able to chat with everyone. And if you click at the bottom of the screen, you click chat, you'll probably see a bunch of numbers there because there has been folks rolling in telling us where they're joining from. So if you haven't done that yet, click that chat button, select to everyone, tell us what your name is and where you're from. And, you know, if you're excited about anything, this is a great time to chat with other folks who are joining us. And right now we have, we've got 150 folks who have joined us so far. So we've got a lot of pollinator folks here. But as we are going through, we are going to run through a lot of information today, and our speakers are going to be talking about their different project and program areas, but we're saving all the questions to the end. So if you have a question, please don't put it in the chat. Please put it in the Q&A section. Charlotte is going to be monitoring that, and there may be some things that we can answer as we go along, but we'll save all those to the end and answer those in front of everyone so everyone can get the information. So we are recording this, so if we run long or if you need to pop off or something happens, it is being recorded. We will post it on the Extension Master Gardener Volunteers of North Carolina YouTube. If you are joining us, you already have the information. So everyone who registered will get all the information after the fact, after it's all posted on YouTube. So it'll probably be sometime next week, but it will come at you, so don't worry. So today, we are trying to accomplish a couple of things. So I did say that this is the third annual Southeast Knowledge is Pollinator Power webinar. And the first kind of goal is to get everyone excited about the Great Southeast Pollinator Census, which we'll talk about in a little bit. But also at the same time, learn some really cool information about pollinators and pollinator gardening. Our three experts who are joining us today are joining us from a lot of different parts of pollinator conservation and have lots of really interesting things to share with us. So I'm really excited to have them all here today and share their information and their knowledge with you guys. Some of y'all, whenever everybody registered, I asked if people were aware of what the census was or if they had participated in the census. And surprisingly enough, about 70% of the people who registered said that they were aware of the census. But if you are, of course, if you are, I forgot what I was going to say. 70% of the people had heard of it, but another 30% had never done it. And then only about 30% of the people who are joining us had ever even done the census. So if everyone here today does one census, we'll have already increased double our participation. And then also let you know about some of the resources that are available to help you on your census journey, whether you choose to participate on your own, maybe bring your own census event to your community, or you're already planning a census event. And this is kind of the first time we've been able to work one-on-one with each other. So I'm very excited about today. I am with NC Cooperative Extension, so we're always trying to share information about who we are. And our mission is to extend research-based knowledge to all North Carolinians, helping them transform science into everyday solutions to help them improve their lives and grow our state, which is such a cool thing. I was talking with my colleague the other day, and we're just essentially professional nerds, helping other people access knowledge to help them with whatever they're doing. So, everything from a lady growing an avocado from a pit that she saved all the way up to a row crop farmer trying to improve their yields and fix, what kind of fertilizer they're using. So we are here to serve everyone in North Carolina. So I think that's very cool. And if this is the first time you've ever heard of Extension, there is an Extension office in every state or every county in North Carolina and the Eastern Band of Cherokee Indians. Please go visit your Cooperative Extension office. We would love to see, we love to hear from folks. So you can actually look at your local county center at this website, or you can just throw it into your Google machine and it'll pop up. I would be remiss if I did not mention the wonderful resource that is the Extension Master Gardener program, because a big part of the success of this program launching in North Carolina has been the mobilization of the Extension Master Gardeners and their ownership of this program across the state. So their goal is to connect people with the benefits of gardening through research-based information, educational programs, and community outreach. And these are the folks who are helping folks every day in your local community, maybe whether you're aware of it or not. And so I am very fortunate to have some excellent Master Gardener volunteers in Lee County, and we've got volunteers in more than 80 counties across North Carolina. So there's probably one in your community right now. And here's our wonderful speakers today. I'm going to read their bios later because they're awesome, and we've got so much ground to cover. But Nick, thank you very much for joining us all the way from California, but also all the way from the UK. So thank you for joining us today. Dr. Hannah Levenson here at NC State, and she's joined us the last 3 years. So if you've joined us in the past, you've seen her before, but she's always a wealth of knowledge. So glad to have her here. And Doug Rous is here joining us from the coast. We appreciate you joining us and looking forward to learning more about the crystal skipper. And all of you are all here. This is a map of everyone who is registered here in North Carolina, as well a little bit of a shout out from some of the states. I did want to ask you, Charlotte, is the New Zealand registration, is that your family? That's so funny.
Not that I know of.
I was very shocked to see New Zealand and Maine. So, but always happy to see everyone who's here. But and folks who are joining us, if you have friends or family or know of Master Gardener volunteers in the counties that are gray, please let them know that the Great Southeast Pollinator Census is happening next weekend, and it would be wonderful to have at least one count in every county in North Carolina. That's like my 5-year goal. And finally, without further ado, I'm so happy to have Becky Griffin joining us today for this webinar to say a few words.
All right. Well, thank you, Amanda. I love the fact you picked pictures of us where we're truly out in the field and, having that experience. Well, welcome, North Carolinians. I'm so excited. This is the 3rd year you all will be participating. The Great Southeast Pollinator Census started as the Great Georgia Pollinator Census in 2019. So we are overall having our 7th year. I want to thank you guys for your enthusiasm about pollinators. I know Amanda and her team have done an amazing job getting you guys geared up, giving you the information that you need to be successful. And I'm looking forward to that weekend. So it's not this weekend, it's August 22nd and 23rd. It is truly a Southeast Pollinator party. Everybody's going to be out learning. We're going to be talking about neat gardens and generating data. But the most important thing I want you to get from from today is your actions do matter. How you garden truly matters in for these beneficial insects and the eco services they provide. Your counts do count. They matter. Whether you're counting as a group, whether you're counting at home, whether you're counting at a school, it is not just a feel-good project. It is a project that truly matters. So thank you to Amanda. Thank you to all the scads of Master Gardeners. We have that same wonderful group in Georgia as well. Thank you to the panelists, and thank you for willing to be part of this really important initiative. So Amanda, I'm going to hand it all over to you and your great team, and I look forward to it.
Thank you, Becky. We really appreciate all you do. And folks who have been to the Great Southeast Pollinator Census website, that is Becky all over the place. So she has been pulling those resources together with her team and is eternally grateful for that. She set us up for success. And to kind of piggyback on what she's saying, I want to do a few slides because I know our panelists are going to go into this a little bit in their own kind of way, but I want to remind folks of why we should care about pollinators. You're probably joining this webinar because you care about pollinators, or maybe somebody you know cares about pollinators and they said you should watch this. But as somebody who's kind of in it every day, who's learning about it and kind of working with folks who are interested in pollinators, it's just kind of like, yeah, you should care about pollinators. Why don't you? And so I really want to remind you who may be like, yes, I know pollinators are important. Remind folks that this is an opportunity to remember that not everyone knows, and you could be the person that helps change somebody's perspective on the importance of pollinators and what they actually do for our ecosystem and our food system. So don't forget to remind folks. There is a lot of hype around pollinators right now, and Hannah's going to talk a little bit about that. You may have seen some of these initiatives come across your Facebook or Instagram or whatever, maybe your email. Maybe some of you guys are part of these programs, and they have done an excellent job of kind of raising awareness to the general public about the importance of pollinators. And so but there's still more to learn and there's still more folks to engage. And here's just a few things kind of rattling off and piggybacking on what Becky said. Why should we care about pollinators? We all like to eat, and we all like to eat a lot of different kinds of foods. We have a lot of different kinds of foods to eat because of the ecosystem services and pollinator services that pollinators provide. With that kind of being said, our economy, in North Carolina, we have I think $110 billion agricultural economy in North Carolina. Agriculture, a lot of our food crops are wind pollinated, but blueberries, blackberries, if it's a berry or a fruit, we need animal pollinators. So, and without those, we would not have $110 billion economic boost. So on that, that's very human centric, but just on we like the earth to support all life. Pollinators are incredibly important for functioning ecosystems, because even if we don't eat it, fruits produce flowers and fruits and seeds, and pollinators help with that life cycle of plants, and without that they would die off. So we like functioning ecosystems. We are seeing a decline in different pollinator populations across the world, and that's kind of a different story depending on what kind of pollinator it is. But we do know that overall there has been a decline in pollinator and insect populations around the world. And so us doing our little piece and our little patch makes a huge difference. And then also, many of y'all are probably here, are gardeners. It really is very fulfilling when something that you plant is visited by a bee or a butterfly or some sort of animal. And it's not just you enjoying it, but it's nature enjoying it with you. And the National Gardening Bureau did a survey about 10 years ago, asking people what they like the most about their gardens, and that connection with nature and that sense of interconnectedness beyond themselves was one of the top rated things that drew people to gardening. So whatever you get from that, it is important for our health and safety and happiness. And also the future. This is my daughter. I want her to see all the cool things in the world. So we need those ecosystems to continue to function. So all of you, as part of participating in the Great Southeast Pollinator Census, you are working through a community science project. This is a little flyer I made for our pollinator garden, trying to just promote different things that people can do in our extension demonstration garden here. But there's lots of reasons why folks do this. So it is a great way for engaging folks. If you have an outdoor or a garden or a conservation-minded mission within your organization or just your personal one, community science is a great way to engage with folks. It's also, for scientists, a really easy way to get a lot of data, and Hannah's going to talk about why that's so important for her. It does help with now that you can enter your data through a website. You don't have to send in your paper forms, and I'm hopeful in the future we'll have a Great Southeast Pollinator Census app. Really hopeful about that. And whenever we've got a scientist at the helm, we know that data can be trusted, and we're going to talk about what the protocol is to make sure that whenever the data comes in, we can trust that it's describing what it's supposed to be describing. And when volunteers get engaged, it's most of the time, it's not a one-time thing. People get invested in a project, and they get invested in the mission and the elements of that program. So it's a great way to connect with people for a long period of time. And there's lots of different ways for people to engage. You know, I have iNaturalist, Bumblebee Atlas, and the Great Southeast Pollinator Census here because they have different levels of complexity. So iNaturalist is fairly simple. You see a plant or an animal, you take a picture of it and you put it on the website, and bam, there's a little more nuance to it, but that's pretty much what it is. The Great Southeast Pollinator Census is a fairly simple community science project with a 2-day window, and there's a lot of community support for that. But the Bumblebee Atlas, and Becky's a collaborator on this as well, but that's a very complex community science project, but it is incredible the work that they're doing and the data they're generating. And I was very excited to see, Nick, and you're on the West Coast, but that's a project through the Xerces Society, and they actually described a completely new bumblebee in, I think, Washington, because of the work that they've been doing through their Bumblebee Atlas on that side of the world. So who knows, maybe that work in the Southeast, we're going to show that too. So the Great Southeast Pollinator Census, those of y'all who do not know what it is a community science project, and it is across the whole Southeast. We're very excited that Alabama is joining us today, so in 5 states. It is next week, August 22nd and 23rd, next Friday and Saturday. That is when the census is. We're doing this just to get everybody jazzed up. And I love the census because it has 3 really simple but very impactful goals: to create sustainable pollinator habitat, by educating gardeners about using plants that provide nutrition for our pollinators while handling our summer droughts and do not have disease or pest insect pressure. Now, obviously, everybody who gardens knows that, well, this year is really crazy with how wet and cooler August has been. But hey, gardening is a new experience every year. The second part of that goal is to increase the entomological literacy of our citizens. So entomology is the study of insects. So we want folks to just look beyond, is that a butterfly or a bee or just a bug, and actually get to know what those different insect types are. And then finally, to generate useful data about our pollinator populations, so we can begin to spot trends and see how pollinator populations are affected by weather and how honeybees influence native bee populations. And Becky is, after our trip to the UK, one of the places that we went to was the UK Palms, which is at their Center for Hydrology and Ecology, which is kind of like the equivalent of our Environmental Protection Agency. And they do a very similar project to the Great Southeast Pollinator Census. And they put out a report every year, and we're hoping to be able to produce a nice report for the census in the future, being able to tell some of those stories that we've been able to see. But before we put that out, there have been journal articles and studies and publications made using the data from the Great Southeast Pollinator Census, mainly when it was the Great Georgia Pollinator Census, because they've got almost 7 years worth of data. And all of these, if you want to read these articles, they are available on the main Great Southeast Pollinator Census website. So please check it out. This is the main website here, gsepc.org. So we've got, and here at if you need some North Carolina specific information, it's go.ncsu.edu /GSEPC in NC. There's a lot of North Carolina specific information there. So you may see both of those, but they're both great resources. So who can do the census? Anyone in North Carolina, Florida, Alabama, and South Carolina, and Georgia. So even you guys joining us here, if you're not in North Carolina, but you're in one of those other 4 states, you can do it. If you got family visiting to your place, to your home here in North Carolina or one of those 4 or other states, they can do it too. So anyone who is present in these 5 states on the 22nd and the 23rd can do it. So everyone all the way from children all the way down to farmers, businesses. We have a few governments that are doing it. So anybody who's here can do it. And it's pretty, where do you do the census? It's anywhere there's a plant that has pollinators landing on it. It doesn't have to be like a really active plant, although some people really enjoy the thrill of watching clouds of insects visit mountain mints, but it can be any plant that's in flower that has a pollinator landing on it. So any of these places, and you don't have to do the census with a large group. You can do it on your own. The data sheet is, you can download it yourself, print it out, and do it in your own backyard. I also know that if you aren't part of Cooperative Extension, there are some extension offices in North Carolina that are offering to print out the data sheet for folks if they're not able to themselves. I'm not saying that every extension office can do that, but please reach out to your local Cooperative Extension office if you need some help with that. Some of them are offering that service. We also have 35 Extension Master Gardener volunteer demonstration gardens in North Carolina. Not all of these are having census events, but these are great places if you're near them that you can go and count if you don't have any plants in your own yard or you just need a different place to go. Here's a map of all of them, and they are free and open to the public year round. This is not an up-to-date map, but if you don't want to count alone, we've got almost 80 events across North Carolina that are happening that are open to the public, literally mountains to the sea. I do know that because I have seen events come in from the mountains to the sea, folks. But so if you're in one of these counties, please consider checking those out. So here is the data sheet. So there are 8 insect groups. Half of them are bees, but that's really important to know. So we've got carpenter bees, which are most folks know as a little bit of a pest, but they are very important pollinators. Bumblebees with their fuzzy bodies, which Hannah's going to talk a little bit about. Honeybees, which are Apis mellifera, that's one type of bee. And in fact, it's one of the least counted insects in our census event. So I was looking at the data. Small bees, which is everything that isn't one of the other bees, and because typically the other bees tend to be smaller than those. Wasps, they usually have a skinny waistline and they don't have hairs or a lot of hairs. Somebody called them the Cadillac or the Chevrolet of the insect world. I think that was Matt. Flies, so they usually have very small antennae and big eyes. And then our butterflies and moths, if it looks like a butterfly or moth, it's probably one of those, so don't overthink it. And then any other insects that have landed on the plant. And if all of this is not making sense, or you're just like, Amanda, this is the first time I've seen this, I have no idea what you're talking about, you can watch this webinar. This was a webinar we did last month, and it is a phenomenal, I'm really proud of Hannah and Matt for helping us with this. So Hannah did a breakdown of all the bees, and Dr. Matt Bertone from the Plant Disease and Insect Clinic here at NC State did a breakdown of the other 4 insect groups, and we really delved into it. And there's a little bit of a pop quiz at the end that was very insightful. So highly recommend if you need a little bit of a primer, this is a good one to watch. So once you got your data sheet, you got your plant, you're going to observe it for 15 minutes. You're going to put a tally mark every single time you see each insect land on the plant, even if you think it's the same one. Statistically speaking, you may not know it's the same one. So Becky has worked that out with the statisticians to to clean that up. So just every time an insect lands on it, works. At the top of the data sheet is a little QR code, and on the census weekend, if you scan that little QR code, there will be a button that says input your data here. It's a very quick online form that you just put your data in, and I think from start to finish, for me, it takes a little under 30 minutes. It almost takes just as long to set your chair up and put your data in as it does to actually sit there. And I find folks, after they've sat there for 15 minutes, you know, in our like crazy world, 15 minutes kind of seems like a long time, but actually people self-report that they feel much calmer and much more connected to their garden and their community. So just from a mental health standpoint, it's a great mental health break. So anyways, if you do choose to monitor a very active plant, try to choose a much smaller section to watch. And you know, maybe pair up with somebody, but really only watch whatever you can mentally keep track of. So if it's something this big, because I know mountain mints can be very, very active, whatever you yourself feel confident keeping track of. So this year I wanted to do a shout out. Oh, I didn't change my picture. We are doing a little bit of a collaboration with the UK Pollinator Monitoring Scheme, our colleagues over at the UK Center for Hydrology and Ecology. And so if you, on the census weekend, if you have an oregano or a marjoram flowering in your garden and you observe it, please, when you upload that data, we plan to share that data with our UK colleagues. They have a little bit more a different protocol than we do. They have a selected list of plants that they ask people to look at, and it just so happens that oregano is one of those, specifically things in the genus Oreganum, which is oregano and marjoram. So I know that I've got 3 different oreganos that will be in full flower that weekend, so I'm very excited to have that other element of collaboration with this. The 2025 counting sheet is available on the main website right now. It's right at the top, super easy to get to. If you can't find it, please email me. We have, Becky has created these wonderful pollinator guides and descriptions. So if you need something to put on your outreach table, or you need something to print out to take with you to the garden to identify these different insect groups, this is already available free for download. And she's done a wonderful job to create the census materials in Spanish, and they are always, and they're also all available for free on the main Great Southeast Pollinator Census website. I'm hoping, so Becky, I haven't even had a chance to tell you this yet, but I'm working on trying to get your trifold into Spanish as well. So very excited to have that available to our Latino community. We do have some official, some people really like the swag, so Becky's wearing the Great Southeast Pollinator Census hat that you can get through UGA's website, but you can also print your own I counted stickers if you're really into that, and you can create a certificate. So if you go on the main census website, there are buttons for both of these. And I'm very proud to say we also have some North Carolina specific stickers created with the insect mascots. And for folks who run census events of their own, thank you to the Durham County Extension Master Gardener volunteers. You can now print whole sheets of these on Avery 5294 paper. There's all sorts of resources on our main website, but there are specific resources and guidance for different groups that may be participating in the census. And so if you go to this website, you can learn more about how to build momentum, some activities you can do, and some resources we have available to help educate your participants. Grab and go. I'm also really happy to work with Charlotte. She puts this Plant These for Pollinators series together, and you can look at the archive of that on the NC State website as well. It's kind of like a plant this, not that kind of thing, and it's a really good resource. Within Extension, so 4-H is part of Extension, and here in North Carolina, Becky and UGA have some incredible curricula as well around pollinators that's available on the main census website, but we have a link to our North Carolina some North Carolina curricula that is aligned with North Carolina Common Core. So if you work with these groups, these are available through your local cooperative extension office. There's a great Facebook group out there. So if you are on Facebook and you haven't joined the Southeast Pollinator Census Facebook group, you need to get on there. Becky posts daily. People post photos from their own gardens. And I know on the census weekend, it is a crazy time on there. People are posting what's going on in their events. So if you want to know what's going on in pollinator land in the Southeast, you need to be on the census Facebook group. So hope to see you there. I'm going to go really quickly through this. I know I'm droning on and we need to get started. But going back to what Becky said, real participation, real data, your counts matter. So in 2024, we jumped from 66 counties up to 78 counties across the state in North Carolina that counted. We almost doubled the number of counts that we had across the state, almost doubled the numbers of insects counted. And I have the top 10 plant genera that were surveyed posted on our website. But some really common stuff that folks probably have in your garden that you may not have realized are pollinator magnets. And I'm really proud that we have had folks from the mountains to the sea, from tier 1 to tier 3, so rural to urban counties. And in 2024, the top 5 counties, Forsyth, Durham, and Cabarrus and Cleveland, those are urban, suburban counties, but Transylvania County is in the mountains. It's a rural county, and they were very active. So you don't have to be in a highly populated urban area to make a huge difference. So everybody's count counts. We have increased the number of events, and I'm really excited to see that we've got so many events happening across the state. So please check out and join a local event. The other thing that Becky talks about is trying to increase sustainable pollinator habitat. So last year, 80 new pollinator gardens were created because of the Great Southeast Pollinator Census, and all of them, again, from the mountains to the sea. So we're seeing a lot of engagement. This is the data that we collected from 2023. So lots of bumblebees and moths and small bees. And that's very similar to the data that we got in 2024 in terms of bumblebees and moths. But we actually saw less small bees and more wasps. I want to note that honeybees are still the least counted insect in both 2023 and 2024. So think about that. A lot of folks only know about honeybees as pollinators, and we have this whole group of insects that are doing the lion's share of the work. So you guys going out and learning about the Great Southeast Pollinator Census can enlighten folks on all these other really important pollinators out there. So my challenge to you as you're listening to our speakers is, what can you do in your own backyard or your community for the census and for pollinators? Okay, I'm done talking because you guys probably came to hear more from these folks. So guys, I'm going to read through all of their bios because we have quite the pedigree that has joined us today. And then Nick is going to go first, Hannah is going to go second, and Doug's going to bring up the rear here. So Dr. Nick Tu is currently working as a postdoctoral scholar at UC Davis, studying the ecological restoration of solar parks. So solar energy, y'all. Prior to moving to the US in 2024, he conducted a PhD at the University of Bristol in the UK, focusing on plant-pollinator interactions in urban landscapes, and has also held research positions for 2 UK charities, the Royal Horticultural Society and the Royal Parks. In his free time, he enjoys being out in nature, especially hiking, camping, and trail running in the mountains. And he's got some incredible mountains up near him. Dr. Hannah Levenson is a research scholar in the Department of Entomology and Plant Pathology at NC State University and head of the Specialty Crops Integrative Pest and Pollinator Management Lab. Whew, it's a tough one. She is a community ecologist researching how to mitigate human impacts in the environment. Her research focuses on mainly making pest management more sustainable and protecting pollinators, especially in agriculture. She is a bee expert with experience working with honey bees and native bees in both the US and in international settings. And finally, Doug Rous is a biologist and outdoor enthusiast from Apex, North Carolina, with outdoor interests ranging from scuba diving to wildlife photography to herpetology. He received a Bachelor's of Science in Biology from the University of North Carolina at Chapel Hill in 2018, and has since worked with Alocid fish. Is that right? Yes, I did practice. Alocid fish, colonial water birds, and the crystal skipper, which is what he's going to be talking about today. So without further ado, Nick, I'm going to mute myself and take it from there, man.
Sweet, thanks very much. Okay. How are we doing on sharing? All right, can you all see the presenter view, okay? Is that working? Sorry, everyone's muted. Just want to check the screen's okay.
I can hear you. Yep.
Okay, and we can see the
Can see you,
presenter
can
view
see.
without the notes. Okay, awesome. Well, thanks so much for the introduction. As Amanda mentioned, yeah, I'm out on the West Coast in Davis, California right now, originally from the UK. So this is going to be a very broad overview to a subject which is relevant kind of across all those settings, which is pollinators and their food supply, and talking particularly about what we can do to feed pollinators in our yards and our gardens. I look forward to hearing some more like North Carolina specific talks later. So I thought I'd just start off by just briefly introducing who I am and why I'm qualified to talk to you today about pollinators. So I grew up in the southeast of the UK, a real kind of passion and fascination for nature, getting grubby, picking up toads in the garden and all that kind of stuff. And that really helped to shape me into the ecologist that I am today. I went off to Oxford to do an undergrad in biological sciences, getting a really broad introduction to everything, really, from like genetic and cellular level up to ecosystems, starting to get really interested in insects and invertebrates, doing a project on spider webs. Then my master's, I started to get more interested in pollinators specifically. So I did a project looking at the effect of pesticides on bumblebees, using a really cool kind of RFID radio sensor setup where we tagged bumblebees and watched them moving between artificial feeders. Then, as Amanda mentioned, I was in Bristol for a few years doing my PhD. This focused on urban landscapes, particularly focusing on people's gardens and how we can feed pollinators and the nectar supply of different plants. I did a short position for the Royal Horticultural Society, which I think overlaps a lot in terms of some of the Master Gardener work that has been going on in North Carolina. We were looking at a pollinator-friendly planting list that I was helping to review, and also doing a lot of pollinator census work. And then I worked for the Royal Parks, which was kind of putting a lot of this knowledge into practice in public green spaces, looking at how we can find some sort of balance between people and nature within London green spaces. Now I'm over in Davis, California, doing a 2-year postdoc looking at ground-mounted solar installations and how we can manage the land in and around panels to more better suit pollinators and other wildlife. So pollinator diversity, I'm sure everyone on this call, and I think we have something like 200 on today, know about bees and probably about hummingbirds as well, given we're here in the States. But there's actually a huge diversity of insects that pollinate and other wildlife. So we've already touched on things like moths and butterflies, flies, beetles, but even bats, and some really sort of perhaps surprising examples of pollinators like primates, like this lemur from Madagascar, or even geckos and lizards pollinating in certain situations. So a paper came out last year showing potential wolf pollination by Ethiopian wolves over in Africa. So this shows an Ethiopian wolf drinking nectar from a red hot poker flower, and in the process getting its muzzle completely covered in pollen. If it goes on to another red hot poker flower and covers that one in pollen, then it's bringing about pollination. So I'm not sure we're going to necessarily talk about wolves as being particularly important pollinators in ecosystems, but the point I'm making here is anything could be a pollinator, but certain groups are kind of much more reliably pollinators because they visit flowers much more frequently. So by far and away, the greatest diversity of pollinators is the insects. So 99% of the estimated nearly a third of a million, or more than a third of a million pollinator species that have been described belong to just 4 insect groups. So these are your moths and butterflies, the Lepidoptera, beetles, the Coleoptera, bees, wasps, also including ants and sawflies, the Hymenoptera, and then flies within the Diptera. So the majority of this talk about pollinators' food supply will consider insects, and particularly the 4 in these groups. So what do pollinators eat? Well, like everything in biology, it depends. So are we talking about larvae and adults? Are we talking about differences between males and females? Are we talking about obligate versus facultative flower feeders? So this is some scientific jargon. Obligate, basically things like bees, they rely exclusively on floral resources, so they have to visit flowers for their food. Others, like yellow jackets, for example, they'll regularly visit flowers for nectar, but they'll happily make do with fruit or other sources of sugar. So this image on the right here, and I should say all the images here that don't have photo credits, I've taken on my various travels. So this one on the right here was just with a cheap macro lens attachment for a phone in the south of England, showing an ichneumoniid wasp parasitizing a caterpillar of the cinnabar moth. So both of these are pollinator species. As adults, the ichneumoniid wasps will feed on nectar from flowers, but as larvae, they'll slowly devour the inside of this moth caterpillar while it's still alive. And similarly, this moth, the cinnabar moth, is a flower feeder and a pollinator, but as larvae, it's feeding on ragwort, which is a toxic plant. So when we think about feeding pollinators, we need to consider, have we got food for them as larval stage and also food as adult stage? Males and females can often differ. For example, in hoverflies, where often the males will feed only on nectar as a sugar source just to keep them flying. Females need protein for egg development, so they might need to take pollen as well. Which brings me on to nectar, which is obviously one of the two main floral resources that pollinators are feeding on as adults. So as I'm sure most of you know, it's a concentrated sugar solution, something like 20 to 60% sugar by mass, which just by reference is like taking a cup of tea or a cup of coffee with 94 spoons of sugar, 60%. So it's incredibly concentrated sugar solution. It can have some other trace quantities of different compounds like amino acids, salts, vitamins, sometimes even toxins. So rhododendron nectar, for example, has toxins that are disproportionately toxic towards honeybees and solitary bees and much less toxic to bumblebees. Bumblebees are also much better pollinators of rhododendron flowers. So this could be a way in which the plant is evolving to try and bias the pollinators that visit it to the ones that are more likely to pollinate it. So we've probably all kind of grown up thinking about plant pollinator mutualisms as this lovely relationship in which both parties want the same thing. But of course, like many things in nature, there can be examples of cheating, there can be examples of subterfuge, plants poisoning pollinators, finding their way to steal nectar from flowers, which I'll talk about in a minute. So a huge world of complexity going on. But typically nectar is largely, with some caveats around toxicity and things, it's largely a simple sugar solution, very rich in energy for flight and metabolism, can also be a source of water, a medicinal source. So there's examples, for instance, of bumblebees taking nectar from certain flowers when their colony is infected with certain parasites as a way of self-medicating. And of course, it can be stored as honey in the case of this honeybee. So the purpose of nectar really is attracting pollinators, and in some situations, pest control. So extra floral nectar, so nectar that comes from stems or leaves, can be used to attract ants as a way to prevent against caterpillar herbivory. So most flowers are secreting just a tiny amount, a few microliters a day of nectar. And there's an example on the right in that photo of a comfrey flower with a little capillary tube in, which is how I'm extracting and measuring nectar. So that's probably something like half a microliter that's been kind of sucked up into that tube. And this quantification can allow us to come up with some sort of quick and easy statistics, like 10 strawberry flowers providing a bumblebee with the energy required to fly 1 mile, a jar of honey requiring something like 1 million rosemary flowers. Obviously, a lot of kind of assumptions made in calculating these statistics, but these are the kinds of calorie level statistics we can start to use to quantify the relative benefits of different plants for different insects. So access to nectar varies a lot with flower shape. This is a really important factor. So we have the difference, for example, between more open flowers, something like a sunflower, accessible to a whole range of pollinators, versus more tubular flowers, something like this salvia on the right. And then correspondingly, you'll get longer and shorter tongued insects. So hummingbirds will have a very long tongue that can happily reach down to the bottom of that salvia flower to access the nectar. But hoverflies, for example, wouldn't be able to get in. But of course, as always, there's a way around this. So there's nectar robbing, which some of you may have observed on your census counts. So this is a carpenter bee that's decided to just bypass the legitimate way to access nectar and just drill a hole through the back of the flower. So that way it's accessing the nectar, but it's not actually bringing about pollination of the flower. So another example of kind of cheating within this plant pollinator mutualism. And of course, horticultural cultivation can affect flower shape. So if you have things like dahlias, some zinnias, roses, flowers that can be bred into multi-petal double varieties, that will often impact the accessibility of the flower or the quantity of floral resources. So again, another really important factor when selecting plants for pollinators to typically try and include more wild type varieties, ones that are less horticulturally modified. So pollen then, in contrast, is the other main floral resource. So it's a powdery solid with grains that are somewhere in the region of 5 to 200 micrometers long. This is a photo I took from Arizona in April, showing an opuntia prickly pear flower with a scanning electron micrograph of a pollen grain. So just by reference, a typed period on a printed page is something like 300 micrometers. So pollen can be up to 60 times smaller as a single grain. And it's a major source of protein, in contrast to nectar, somewhere in the region of 10 to 60% protein by mass. Also a really important source of lipids, so lots of fats and kind of important compounds like that, carbohydrates, vitamins, very nutritionally complex. And as a purpose, it's partially for plant reproduction. So of course, transferring pollen from one flower to another is how plants reproduce sexually, but sometimes also as a pollinator attractant. So sometimes flowers are producing much more in order to bring in pollinators, knowing that they'll take some proportion of that and eat it themselves. So as I mentioned, it's nutritionally much more complex than nectar, and therefore insect pollinators are usually much fussier about pollen than nectar. So this leads to something called a specialism, where you get bees that are monolectic, oligolectic, or polylectic in their pollen preference. So getting quite jargony again here. Mono, meaning one, means they'll only visit one species of flower for pollen. That's quite rare, but it does exist. Oligolectic, which is much more common, are bees that will only visit a handful of flowers, and typically they're quite closely related, so the same genus or the same plant family, because they'll share a lot in common in terms of the chemical profile of the pollen. And polylectic means things like your honeybees and your bumblebees. They just want a whole variety of different pollens. They're not too fussed about specifically what species are comprised. So example on the right here is a bellflower blunt-horn bee from up in Scotland, and this is a campanula specialist. So this will only take pollen from the bellflower genus. Doesn't really care if that bellflower is native or not, but it has to be from the campanula genus. So I've discussed, or mentioned briefly at least, what, where, how much, and how to access food for pollinators. The other really important factor is when. So this bumblebee on the left here is a queen. It's a queen tree bumblebee in the UK. It's just emerged from months of hibernation on February 20th. Now, if any of you have been to the UK in February, it's not a lovely place to be. It's very bleak. There's very little in flower. If this bumblebee queen emerges and there's no nectar to give her energy to fight off the cold or pollen to allow her to produce eggs for the new colony, then she will die. So it's important that these crocuses are around in February. And this graph on the right here, which I think is, you'll be pleased to hear, is the only graph I've included in this presentation, is some work that me and a colleague did in my PhD, where we quantified the profile of nectar, so the quantity of nectar produced by different habitats through the year. And we compared some more rural, semi-natural habitats, you might say, to urban gardens. And what you'll see is woodland, hedgerow, pasture, and field margin have quite a boom and bust nectar profile. So nectar kind of peaks in late spring or in summer. Maybe there's a few separate peaks, but there's very little in March, April. There's very little in October, November, and there's often kind of gaps during the year. In contrast, gardens, much, much more smooth, continuous profile of nectar. So there is more in June, July, but there's also quite a lot in March and October. And in a way, this isn't surprising. It's a variety of factors. It's many gardeners managing their gardens differently. So there's always one that has something in each time of year. A lot of gardeners being actively interested in providing flowers all year round, whether that's for wildlife or for their own aesthetic benefits. Urban heat island effects, potentially extending flowering seasons, different varieties, different species, some of which are non-native, allowing this flowering season to be extended. So gardens really seem to be incredibly important for providing pollinators with food at certain times of year. So this brings me on to the final section of the talk, which is what can you do? So very simply, quantity of flowers is important. So packing your yards with as many flowers as you can fit in, thinking perhaps about not having a very large, short area of mown lawn, or at least mowing less frequently and embracing that untidiness where those flowers can be allowed to grow up and actually flower. Obviously avoiding herbicides as much as possible and pesticides and all toxic chemicals that are going to be damaging to native wildflowers or pollinators themselves. And then trees, shrubs, climbers, these are really good ways of packing in a load of flowers in a smaller space because of the three-dimensional access. Diversity of flowers is obviously incredibly important as well. So planting a wide variety of species, different flowering shapes, different types of nectar or pollen, different flowering periods. So you're trying to extend that flowering season all year. This image on the right here just shows the UC Davis bee haven. The main two flowers you can see are California poppy, the orange one. That's a pollen-producing plant, no nectar, open and accessible, really important to a lot of bees and hoverflies. And then the purple one is a salvia, maybe Salvia nemorosa, something like that. That's more of a nectar specialized flower. Only certain slightly longer tongued insects will be able to reach it, and it's providing nectar as opposed to pollen. So just by having those two plants together, you're suddenly able to provide food for a much wider diversity of pollinators and pollinator needs than you would by just having one. But of course, trying to extend that and have as many as you can, flowering at many different times of year is, very important. And the final point is supporting full life cycles. So it's not just about nectar and pollen. It's about nesting sites, which I haven't even had any time to touch on. It's about food plants for caterpillars. It's about often freshwater resources. So a lot of hoverfly larvae, for example, will start out their life in ponds, deadwood, particularly for beetle larvae. So I appreciate this is a very UK looking yard, particularly because there's very little sunshine, but you can see a wide variety of different resources provided: shrubs, flowering borders, a longer grass area of wildflower meadow, areas of bare soil, perhaps for nesting, fruit trees. So trying to think about the microhabitat diversity of your yard and not purely about the flowering plants. And in the last couple of minutes, I just want to touch on a really big contentious topic that I haven't got anywhere near enough time to do real justice to. And this is, should we be planting native plants or non-native plants in our yards? So the first point to make here is native and non-native is a very forced distinction. There's many different ways of looking at native and non-native. So I looked up the state flower of North Carolina, which is Cornus florida, a species of dogwood, and that's native to the eastern US and parts of Mexico. Here are three other species of dogwood. One that I saw near Lake Tahoe in California, down in the Southwest, photo Cornus natalii from parts of the US and Canada. Then we have Cornus causa from East Asia and Cornus suecia, which is Swedish, but found all around the northern parts of the world, including Eurasia and North America. So when we think about native status, are we thinking about the country it's from, in which case three of these are native? Are we thinking about the state? So in the case of North Carolina, only one of these is native. Are we thinking about the continent? Are we thinking about the family? Are we thinking about the genus? There are basically multiple levels, I would say, of nativeness. It's not, there's things that are less native than others, if that makes sense. So is it better to plant natives or non-natives? Well, the first thing to say is there's a lot of important kind of moral and ethical reasons why we might prefer to plant non-natives. There's a lot of uncertainty around introducing new non-natives with regard to invasiveness. But what does the actual science say on the pollinator front? Well, nectar, it doesn't really matter. Nectar from one plant is very similar to nectar from another. There's variation in concentration, but there's really no reason why sugar and water has to be particularly different, whether it's a non-native species or not. So this English bumblebee on the right, for example, is very happily drinking nectar from South African agapanthus. Pollen does sometimes matter, because as I mentioned, there's the monolectic and oligolectic bees that often take pollen from a single genus or family. So it often matters if it's native, but it's often about whether it's a native genus or a native family. And then food plants, it probably matters even more. The level of specialism in caterpillars is even higher than pollen. But again, that specialism is often genus or family. Sometimes it goes as far as being a certain native species. So the brief take home is natives do tend to be better, but near natives, particularly same family or genus, are often just as good. As always, diversity of species, flower shapes is key, and avoiding anything with invasive potential or uncertainty around that is really important. So providing a bit of an all-you-can-eat buffet for your yards and your gardens, where not every dish has to be attractive, but there's hopefully something out there for everyone is what we should aim for. So that's all I have time for today. Feel free to email me with any questions, but also I'll hang around for the Q&A at the end. Thank you.
Thank you so much, Nick. Oh, and Hannah set her right up for it.
Yeah, thank you, Nick. That was a really great overview. And I'm going to be referring back to some information that Nick brought up when I start talking about this next presentation. So I want to start with saying we got a broad overview of many different insects that can act as pollinators. I'm going to talk about one of my research projects as an example of why understanding these nuances can be really important when we're thinking about how we want to conserve and protect pollinator populations. So for that, I am going to narrow down and focus on bees, but these concepts can be applied to other types of projects, other insects. Just look at this as one example. So don't get caught up in too many of the details. But the project I'm going to be talking about today is about managing overwintering pollinators in your gardens, thinking about how do we manage our perennial stems when our flowers are done flowering so that we can help support stem nesting insects. And this project was co-led by Elsa Youngsteadt. There's several others that were involved, and I'll share more information about that in a second. But we're going to focus in on stem nesting bees. And the reason why we did this project is because if you're a gardener, I'm sure you've had this thought. You were wondering, I always go in my garden, I always manage it, maybe I trim back at a certain time of year, but is that the right time to do that? So we were getting a lot of those questions as well, and we did what probably all of you would do, and we went to the internet. And if you've done this yourself, you've probably experienced the same thing as us, where there was a lot of different information. Sometimes it was completely opposite from another piece of information we found. So different times of year are being recommended. A lot of times it is people's opinions or something they observed. We weren't finding information that was based on a research project, and we weren't seeing something that was clear and made sense to us. So we decided that we were going to figure this out for ourselves so that we could give recommendations that we felt good about to people about how to manage their garden. So the main question here is, when do we go into our garden and when do we clean it up without impacting the pollinators that we're trying to support by planting a garden to begin with? And like we've talked about already, some of the slides Amanda shared in the beginning, there really is a lot of focus on planting gardens for pollinators right now. You could consider it a pollinator garden boom. For example, the photo on the upper right-hand corner, the Million Pollinator Gardens project, you can just see how many different gardens are being planted. And even though each garden might be small, you can see when you add all of these points together, it actually comes up with a pretty big amount of land. So we are potentially having a really big impact on pollinator populations. We want to make sure we're doing the right thing. And the tough part is, while your gardens hopefully look like the bottom left picture at some point in the summer, right? Like nice, beautiful, your neighbors can appreciate it as well. Eventually, they're all going to senesce, the flowers are going to be done, and it's going to look like the bottom right corner. And that's when less people appreciate it, probably. Maybe yourself, you like to have a tidy look, so you want to know when you can go and get your hands dirty in there. So this is going to happen, and how do we manage the gardens properly? So back to this slide, if we're thinking about what insects are using these stems, as far as bees go, we're thinking about stem nesting bees, but that doesn't apply to everyone. As you got a little sneak peek with Nick's presentation, there's a lot of different pollinators doing a lot of different things. I'm going to orient us in the next few slides, but I want to introduce by saying we now see how diverse bees can be, just the numbers of different species that are considered bees. They can look very different from each other, different shapes, colors, sizes, and I'll tell you they live very differently from each other too. So while that makes them exciting, it makes them very important in our landscapes, that also means it's really complicated and nuanced to work with this group of insects, and how do we know how to support them when they can be so different from each other. So in North Carolina alone, we have about 564 different bee species. And again, they can live very differently from each other, which we'll get into in a second. In North America, there's about 4,000 different bee species, and across the whole world, there are more than 20,000 different bee species. So they all play slightly different roles, they have different interactions in the environment, and we need to keep that in mind if our goal is to support pollinators in general. So the examples I'll give today that we want to keep in mind are different sizes of bees, where they live, how they live, and I'll move quickly through what they eat since we got a really great introduction to that already. So bees can be very different in their size. I want to say that when a bee emerges as an adult, it is fully grown. So if you see a small bee, it's a small bee, it doesn't need to grow anymore. So the smallest bee in the world is Perdita minima, it's 0.08 inches, compared to the largest bee in the world, Megachile pluto, which is 2.5 inches. And there's a bee species that could be any size in between these two. So you can already imagine how a 0.08 inch bee would behave very differently, interact very differently in the environment compared to a 2.5 inch bee. So already starting to see how these differences can happen in the environment. Where these bees live are also different. So when we're thinking about supporting bees, we need to provide them with food, but they also need a place where they raise their young. That's the next generation. And there's 4 broad categories of materials where they do this, where they lay their eggs and make the next generation. So the first category is soil, where a female bee will burrow straight down into the ground. She builds tunnels. Sometimes they can be very complex and extensive. She lays her eggs there. Then there's wood. That could be dead sound wood, or that could be dead rotting wood, depending on the species. We have stems, hollow twigs, and then we have cavity nesters, where there's a large open space and they then fill that with their nest. I want to make a quick point that cavity nesters, the best examples for us in North Carolina are honeybees and bumblebees. Those are very iconic species. We probably know the most about those species compared to many of the other species in North Carolina. But you can see noted at the bottom there, less than 1% of all bee species in the world live this way. Where if you look back at the soil nesters, about 70% of all bee species nest underground. And we know much less about them compared to our cavity nesters. So when we're thinking about supporting bees, there's still a ton of information that we don't have that we need to know and we need to collect good data on. Okay, so then within those nests, bees can also live very differently from each other depending on the species. And I'm showing you a gradient. I'm showing you the 2 extremes. I'm only going to talk about those for time, but just know there's so much nuance in between. I'm just giving 2 examples. So one side of this spectrum here, we have solitary nesters. That is where an individual female builds her own nest, lays her own eggs, collects her own food. She does everything on her own. She does not get help from another adult. Usually they're on an annual life cycle, and so she's laying eggs and then she passes before she ever meets those eggs that she laid as adults. So the adults and the young don't really meet each other. The other side of the spectrum, we have highly used social species. So that's where up to tens of thousands of individuals live together in the same nest. They are working together, they are sharing tasks. Some of them will collect food, some of them take care of the nest. They do have overlapping generations, so eggs are being laid, adults then see those eggs grow up into adults and they all meet each other. These nests usually last for many years, and so already you can see an annual life cycle, perennial life cycle plays a different role in our environments. And then again, the best example for highly used social species is honeybees for us. Less than 5% of all bee species worldwide nest this way. It's very unique in the bee world, where if you look back at solitary, more than 90% of all bees nest this way. They're solitary nesters. And again, we know much less about them, but they're making up the majority of the bees that are out in the environment. That kind of gets back to some of those numbers that Amanda brought up at the census results, where the majority of bees that are out there in the landscape, we don't know as much about. And then really briefly, what they eat. So they all need pollen and nectar, all of these bee species, to successfully raise their young, like Nick was saying, but where they go to get that food can be different. And we learned a lot about those different plant options already, but I just want to show you it is a spectrum where on one side we have specialist pollinators. Maybe they need a specific family, genus, or even species of plant. Usually that results in a close relationship, a mutualistic relationship between that pollinator and that plant species, where that plant needs that pollinator to visit it to have successful pollination. On the other side, generalist pollinators, like we talked about, if a plant produces pollen and nectar, because not all do, But if it does, a generalist pollinator could probably visit that plant and successfully collect those resources and feed it to its young. The caveat here is that generalist pollinators then need to visit a variety of plants to get all of the nutrients in their diet. So when we're thinking about making land management decisions, it's really important to consider all of these differences between bee species. What we do might impact stem nesting bees. For example, in the garden, if we're trimming stems, that impacts stem nesting bees, but it's maybe not going to impact wood nesters or cavity nesters or ground nesters. So we need to think about the actions we're taking, which group that affects, or if there's a particular group we care about, what can we then do to help support those species specifically? So with this project, the groups of bees that are probably being impacted by what I'm talking about are stem nesters, they are solitary, they're probably going to be generalists, and they're more often going to be smaller bodied bees. So keep that in mind when we're talking about this. Okay, so back to this question, this example, we got a question on when do we manage our gardens. This is a really brief graphic about the life cycle of a garden within 1 year. It grows, puts out leafy vegetation, then that plant will flower, things will visit the flowers senesce, and then there's seeds, maybe the birds eat the seeds, and then in the winter, everything's kind of inactive. And this is typically when people start thinking about managing their garden, but when exactly is the right time to go in and manage your garden? So to do this project, we collected samples from 10 different counties across North Carolina. We worked with 9 extension agents, 18 different master gardener volunteers, and 20 different gardens. So really great to be able to get information from all the major regions of the state, and we could not have collected this data set without all of this volunteer work. And with that, I'm going to briefly thank everyone that was involved in this project. So this project was funded by the NC State Extension Consumer and Youth Horticulture Working Group, which Charlotte and Amanda are both a part of. So thank you both for that. We had undergraduates working on this project, and then we had a bunch of our volunteers and extension agents. So thank you to everyone for working on this project with us. Okay, so back to the garden. I talked about this first year life cycle, vegetative, flowering, seed set, and then it senesces. And at this point, if you weren't to do anything, it would repeat this cycle the next year. And so there would be this dead growth from the previous year when the cycle starts over again with new growth. And so the first thing I want to have you all do for when we're talking about this project is to start thinking about the garden on a 2-year cycle. We have the first growth period, and then we have the second growth period where we're following this same stem that grew in the first year, and now it's an inactive dead stem. And that is the focus for our project that we had. So in this section, this is the time period we're focused on in this garden. We're thinking about once the stem senesces, and then it starts growing again next year, when in here are we supposed to be managing the stem so that we're protecting pollinators? And so the first thing we did was we had all of the volunteer gardens trim about 100 stems back to 18 to 24 inches so that we had a standard amount of stems, a standard height to sample from. And then after that, and each of the major seasons, so winter before last frost, spring, summer, and then fall before first frost, they would collect a small amount out of that 100 stems, so about 30 stems in each of these time periods. They would then mail those stems back to us, bundle it by different plant species so we could track which plant species this activity was happening in. And then at NC State, we would receive these packages. We would split them open to see if there was a stem nesting insect in there or not. If there was, we would try to identify what was in there. Sometimes we got lucky and we found a really easy to identify life stage, the adult. If we found something that was not an adult, we would try to rear it in the incubator so that we could then get an identity on it. And overall in this project, we received 68 samples from our gardens, and then counting each individual stem that was mailed to us in that 68 sample set, we processed 2,356 stems, so lots of stems. And I'll be honest, when Elsa and I started this project, we know they're stem nesting bees. Obviously, they're out there. Obviously, they're nesting successfully in stems. But neither of us had actually gone out and looked for a stem nest. And so we were a little worried that we were looking for a needle in a haystack, a 2,356 large haystack, and we weren't sure what we were going to find. But we were really, really excited, and we actually did find active stems in this data set. So I'm showing you a couple of the different types of stems that we found. We found them in many different plant species. We found them all across the state, as you can see listed here. And so we were really excited that we actually could successfully track these gardens and figure out when bees were nesting in these stems. And like I already briefly hinted at, we also did find adults in the stems at some point. So we found many different life stages. All bee species go through the same general life stage. An adult will lay an egg, it then hatches into a larva, then into a pupa, and then again into an adult. So we were able to find all these different life stages in these stems at different points in the garden. So to give you an idea of where we found these, so we had a total of 35 stems that were occupied. Out of our over 2,000, that means only 1.5% of stems were occupied. So even though we were really excited, that number is not the most exciting, but it just shows you that a lot of work is needed to understand these bees and learn more. We didn't know this before, that we would find these stem nests, so even finding 1.5% of stems occupied is a success. That was out of 12 of our gardens, so 60% of our gardens had active stem nests in them, and 32% of our overall samples, so those 68 sample sets, had active nests. We also found some stems where maybe we didn't see active nests, but we found evidence of nests, and if we added that into our total, that's 41 stems, so it bumps it up to 1.7%. So still a low number, but useful information nonetheless. Out of these stems, we found 12 different species of bees and wasps, and they were nesting in 20 different kinds of plants. So we were finding low numbers, but it's a pretty diverse community of bees that we're finding in these gardens, and they're using a diverse community of plants. To give you an idea of the species that we were finding, the majority are Serratina, so those are called small carpenter bees. They're actually related to large carpenter bees, which I'm sure some of you have experience with. Maybe they've nested in some wood structures where you wish they didn't. But they are not they're not destructive like their cousins. They're nesting in these stems in your gardens, but they're just very closely related. We found several different types of leaf-cutting bees. So Hoplitis and Megachile are both leaf-cutting bees. We found them nesting in these stems. We also found yellow-faced bees, Hylaeus, and then we found some solitary wasps as well. And solitary wasps are good neighbors to have in your garden. Like we learned, they will, eat other insects, so they could be providing pest management in your garden, and they can contribute to pollination as well. So they are good neighbors to have in the garden. And then because Serratina was such a large portion of our species distribution, I want to take a second and just give you a little bit more information about Serratina. So they do fall in those general categories I talked about with what to expect in the garden. They are solitary stem nesters, they are generalists, they are small-bodied. You can see they have to fit in this plant stem, so they are very small. And they build their nests similar to how carpenter bees build their nests in wood, where it's this linear structure. They lay an egg on a pollen nectar ball, and then they have a cell wall that separates each of the larvae. So they all get their own little room within this stem nest, and they build these chambers in a linear fashion until they fill up the stem. So just a quick, a little bit more detail on Serratina, because they were such a big portion of the bees that we found. And so if you're a gardener, you probably have been waiting for me to tell you what plants did they use. So first I'll walk through the plant species that we had in our sample set across the top of this chart, and then across the side of the chart, I'll show you those main insect groups that I just showed you in the pie chart. And then the darkness of the color within this chart shows how often we found that insect species in that plant. So the darker, the more often that interaction happened. So you can see Serratina, we found them in Echinacea, Solidago, Vernonia, and Salvia most often, but we did find them in a few other plant species just less often. And then whenever we found a nest, we also took information about the size. So even though all of these insects are relatively small, they do use different sized stems. So another thing to think about in your garden is if there's certain species you want to support, certain kinds of insects, the size of the stem from each plant species can matter. These bees need specific sizes. Okay, so then wasps, you can see we found them in pretty different plants, and maybe there's less diversity of plants that these wasps are utilizing. We also found them in different sized stems, so something to consider. And then because Hoplittus, Hylaeus, and Megachile were less common, I'll show you all of them at once. But you can see altogether the insect community is using quite a diverse set of plants in the garden. And so if you want some more details, here is a list format of these different plant species that were being used in the garden. Okay, so really quickly, I'm coming up on the end of my time. I want to get back to the season question. When is it safe to manage these stems in your garden? So I'll show you through the different seasons where we collected samples. First, in the very first winter, we did not find any nests, any active nests the first winter after stems were trimmed. No activity. Not surprisingly, the spring was the most active. We found the majority of our stems during the spring. We also found stems in the summer and stems in the fall. So we found stems at any point in time after the first winter. And so with that, we actually would recommend completely changing the information we found on the internet. And we would say the only safe time to trim your stems is actually in the very first winter. And then at any point after that, you have the opportunity to be interacting with a stem nest. So again, I'll really quickly skip ahead to, this summary slide. So think about your stems in your garden on a 2-year life cycle. You have the growth period during the first year, and then the nesting activity period in the second year. So we need to be thinking about gardens on a longer term. Only trim your stems in your very first winter. After that, nests are possible at any point in time throughout the year. Our volunteers found that in the second year, stems will naturally start to break down. So if you're worried about the aesthetics, the stems aren't going to be there forever. And trimming stems to begin with does create habitat, so it can be helpful if you're trying to support bees. And then I'll end on this slide, which is if you want more information, we have an extension post, a fact sheet, and a video explaining all of this information. And I will stick around for questions at the end as well.
Thank you so much, Hannah. That's excellent. And a quick shout out to, the folks who have, we did have a pollinator outreach kit giveaway for folks who had let us know by a certain time that they were hosting an event, and your publications were part of that giveaway. But all of those resources are available on the Great Southeast Pollinator Census website as well. Okay, Doug, bring it home, man.
All right, can do. All right, so sharing screen. Share. All right, so hey folks, so I think that while Nick and Hannah, talked about like larger, like groups like bees, and then like nectar in general. My talk's going to be a little bit more specific, and it's going to be on the crystal skipper, which is one of North Carolina's very own butterflies. So I'll just begin by saying something that I think will be, very uncontroversial with an audience like this. Insects are extremely cool. And given their crazy diversity, ecological impact, and biomass, it perhaps no group better embodies Darwin's famous quote, "Endless forms, vast and beautiful." And their importance varies from pollination, agriculture, nutrient cycling, and many things beyond that. But we unfortunately are facing a very, very rapid decline in insects. And that is no less true with Lepidoptera, which is the order that includes moths and butterflies. As you can see from this figure here, this is a survey done of different butterfly species, and 13 times as many were decreasing in abundance as opposed to increasing, which is a real shame because Lepidoptera is an extremely fascinating group. So it's equaled or exceeded in diversity only by Coleoptera, which is the beetles, and Diptera, which is the flies. And to put that in perspective, there are more species of beetles than there are plants. Lepidoptera is not quite that diverse. They're a cool, runner-up, but that still speaks to just how incredibly diverse they are. In fact, they are the most diverse group of herbivorous animals on the planet. But so we're facing this obvious decline in insect abundance. And yet with Lepidoptera, amongst other taxa, we have many very rare, very specialized species. And we lack information on their abundance, on their demography, life cycle, things of that nature. So we need to be gathering data on these species as we to make data-driven decisions as we deal with this in rapid insect decline that we're facing. And so as an example of one of these rare butterflies, we have the crystal skipper, which is the species I work with. So many of you, so it looks like we have a predominantly North Carolina audience. So it wouldn't surprise me if many of you had been to Beaufort, North Carolina. And if you have, you've probably, walked around, that beautiful waterfront that the town has. And if you were looking across the way, perhaps at some of the wild horses on the island across from you, that's the Rachel Carson Reserve. And so we have the Rachel Carson Reserve highlighted there. And then I would imagine many of you have vacationed in somewhere like Atlantic Beach, Pine Knoll Shores, or Emerald Isle. That's the Bogue Banks, which is the big island there in the middle. And then we have Bear Island, which is part of Hammocks Beach State Park. And that is it. That is the entirety of their range. So it's only a 30-mile range. And the species was only officially described in 2015 by a man named John Burns. He named it, so the scientific name is Atriptonopsis quinteri. So he named it for Eric Quinter, who was the lepidopterist who first really took an interest in this species in the 1978. Its name is a kind of a little double entendre. So Allison Leidner at the time was a PhD student studying this species. And this area is commonly referred to as the Crystal Coast. And as you can see with this cute little guy here in the corner, they have these little white flecks on their wings that kind of resemble little crystals. So the name is kind of a little fun double entendre that locals will get. So it's one of only 2 butterfly species endemic to North Carolina. So when I say endemic, I mean found here and nowhere else. So in the same way that, Galapagos tortoises would be endemic to the Galapagos, lemurs would be endemic to Madagascar, the crystal skipper is endemic to North Carolina, and specifically these 30 miles of Barrier Islands. I don't have a picture of it specific, but in this recurring picture you'll see in the background, you'll see its host plant, seaside little blue stem, which is a dune grass that the females will lay their eggs on, and is what the caterpillars will consume, and also what they will build their tents out of. So if you're asking yourself maybe the question, okay, so why is it found here and not over here? Well, in the absence of its host plant, which is a dune grass, the species can't exist. So to go a little bit in the life cycle, this species emerges at 2 distinct periods. So they'll emerge, so one group of them will emerge in the spring, and then another group will emerge in the summer. And so eggs laid during the spring flight, so during that spring, will then emerge that same summer, and the ones laid that summer will then emerge the following spring. So one is emerging after a relatively short period of time, and then the other is actually overwintering and then emerging. And so you can see, so this figure here is from the Burns paper that was the first from 2015 that first described this species. And you can see, right here just how different these instar levels look as they progress. And so when I say instar, so the crystal skipper is an arthropod. And arthropods, go through what's called ecdysis. So that's where they shed their external skeleton, and then will molt and then emerge. And so each time these caterpillars are molting, that's a new instar level. So these little guys with the black heads here, that's an N-star 1, and then the N-star 2, and then going down to the N-star 6s. So our project, this is a collaboration between several different organizations, primarily North Carolina State University, the North Carolina Aquarium at Pine Knoll Shores, and as well as Duke University. It's actually Laura Taylor's a PhD project. So this is Laura Taylor up here in the, no, I'm kidding. No, this is Laura down here. I get them confused sometimes. But this is her PhD project. You'll probably recognize the name Dr. Elsa Youngsteadt from Hannah's talk. So she's the lead PI, and then Carol, Nathan, and David are the other PIs on this project. So you can probably tell, from the amount of like organizations and people involved in this is not a small project. So we're going to be spending 3 years gathering data. So last year was the first year, and then from there, 3 years gathering data on abundance, demography, behavior, and habitat for this species. So conservation objective 1 is, relatively straightforward. We need to, document how many we have now. We need to, look, be able to assess, the current population we have of the crystal skipper, so that, over time we can, monitor those long-term trends. So one part of that is our vegetation surveys. So, going out with a quadrat, which is what Bree has right here. Doing vegetation surveys in the area, as well as looking at nectar resources. So that's part of it. And then mark recapture, which I think anybody who's worked on this project will tell you is the most fun part of it. So here we have a very beautiful female crystal skipper right here. You'll notice that she actually has a marker A8 on her wings. So for mark recapture, we're actually getting our nets, capturing these butterflies, and then giving them a unique alphanumeric code on their hind wings. So for a slightly larger species of butterfly, like say a monarch, you could place a tag on them. But these guys are so small that we actually write on their wings. And so when people ask me what I do for a living, I'll often jokingly say I'm a butterfly tattoo artist. So you can see her right here. And then mark recapture is it's a really good way of estimating population in a specific area, because these butterflies aren't necessarily like, having breakfast in the Rachel Carson Reserve and then having dinner in Hammocks Beach State Park. They tend to stay more local than that. So it helps you estimate abundance in a given area based on how many you were able to, recapture, of the ones that you did mark. And then there's counts, which is where we will, so across its range, we will have these transects. So just these 250 meter like mark strips that we will like walk each time and count the butterflies that we see along there to estimate population. So if you're walking down one of these, one of these transects, looking for butterflies, there's a number of ways you could do it. One that you'll see very commonly in the literature is what's called a pollard walk. This has some definite advantages. One person can do it. Again, it's very common. And so you will be walking down this 250 meters, and then each time you see a butterfly within 5 meters of you in any direction, you will mark it down on your data sheet. So that's great and all, but this doesn't offer the ability for you to estimate like your detectability, because if you didn't see a butterfly like this one right here, then you just didn't see that butterfly, and there's no way of knowing how many you saw versus how many you didn't see. So pollard walks really only give a population index. They don't give a detectability estimate. So it's much harder to compare them site to site, year to year, if you're only using the pollard method. And then, so to contrast that, we have double observer, which is where you'll have 2 people walking down the same transect. Then you'll get over here, okay, so maybe one person saw this butterfly and then the other person didn't. And then, same with the other person, with one of these other butterflies. And so what that can give you is a detectability estimate, because for each person, based on how many, you're able to tell how many they didn't see, as they're walking down, because you can reference that with what the other person saw. And because of that, that can give you a detectability estimate. So and so it's much better for like giving like the population in that area. And then another way is what's called distance sampling. So again, this can be done with one person. And so for distance sampling, as you're walking down the transect, you're not only recording the butterflies that you're seeing, but you are also recording how many meters away it is from you. And so this is operating under the assumption that you will see a butterfly if it is on the transect, and then your detectability will drop the further away you are from, like the further that butterfly is rather from the transect. So you'll be seeing a much higher percentage of the ones that are closer to you, and then you're only maybe seeing a few that are further away. And so that gives you detectability, estimatability, because you're able to look at the ratio of how many you're seeing like closer to you versus further away. And from there, extrapolating like how many it is that it's likely you're missing as you're going through, which again, you're able to like get a much better estimate of the population in the area. Okay, and then so for conservation objective 2, there was a conservation plan created by Dr. Carol Price, who's one of the PIs on this project in 2021. But the thing is, there's still a tremendous amount that we don't know about these species, and so obviously, if we're giving recommendations about like its management, we want to close those knowledge gaps. So the objective is to gather information about habitat suitability, demographics, life history, just so we can have that data and be data driven in our recommendations. Actually, I like this pic. This is actually a robber fly down here in the corner, preying on a crystal skipper. Yeah, I hate seeing them dead, but it is cool seeing other cool little critters like doing what nature does. All right, and then for research objective 1, so we're actually quite fortunate just by happenstance with this project, or just rather the conservation of the crystal skipper, in that we have that 30-mile range there, very small, but just by very fortunate happenstance, there's not one but two state parks actually that fall within that range. So we have Fort Macon State Park on the north of the Bogue Banks, and then we have Bear Island of Hannock's Beach State Park. So both of these areas provide like intact dune habitat, so like relatively undisturbed, like food resources, like availability of host plant, et cetera, for the crystal skipper. But, again, with this being like a major, a place where, people live, as well as a place where even more folks vacation, there's a lot of urban area and much more fragmented habitat that comes with it being an urban area with like large swaths of the island. And so the question really is, how are the crystal skippers utilizing that more fragmented urban habitat? Are they how deep are they going? Are there barriers to them going like in further? And so as we look at our demography, like where they're laying their eggs and things like that, how does that map onto these more like fragmented resources? Is it more the availability of nectar that matters? Is it availability of the host plant that matters? Is it just the size of that more fragmented habitat in general? So that's what we're looking at. And one thing that we've definitely identified is two particularly like important flowers for the crystal skipper. Those are two species of morning glories that just have a tremendous amount of like nectar in them, and then also like relatively high sugar concentration as well. So interestingly, we rarely observe them eat during the spring, but then in the summer they're eaten all the time. So we'll see them eat on a variety of flowers. But I'll say if you're out there during the summer flight and you see a patch of morning glories, it's sometimes like shooting fish in a barrel because they love these flowers here. And then for research objective 2, it's more the demography side of things. So looking at like where they lay their eggs and like the various different instar stages. So some of the ways that we can look at that are, when we're doing our mark recapture, we will catch gravid females, such as this cute little girl right here. She's happily sipping on some sugar water that we've provided for her. And so we'll capture, and when I say gravid females, I mean they are carrying fertilized eggs. So if we can grab them, and then we'll put them in these cages. That's Elsa and Laura working on one right now in that image. So we'll let her go in there. Hopefully she'll lay some eggs, and then we can and then that's a cordoned off area, so we can track those eggs and those caterpillars in there. And the other way we'll do it is we'll be walking along the same transects where we do our counts and our vegetation surveys, and we'll comb through the grass. That I'll tell you that's probably the hardest part of this project, is just combing through those so many blades of grass. I'm looking for these tiny little eggs that they're laying on the host plant, as well as looking for the tents and the caterpillars as well. And so what we're looking at here is are there what phenomena seem to impact their survivorship. Are there if there's a heat wave or something like that, is there like a particular instar stage at which they're much more likely to die from high heat than other stages and things of that nature. So we're aiming to get that demographic information. Next to these cages, we will have instruments that are like continually recording like heat, like UV exposure, things like that. So we're able to compare it with that information to see what is impacting survivorship, and obviously the high heat and like increased like storm severity due to anthropogenic climate change. Those are things that we want to have a model to where we can look at that. And so where do we go from here? So I kind of broke this down into two big categories. So that's as a collective and then as individuals. Things like habitat fragmentation and climate change are beyond the scope of what we can do as individuals. That's a question of political will and grassroots action. But the thing about climate change and habitat fragmentation is these are things that can really compound on each other when we're talking about something like the crystal skipper. So one thing that's like well established in the literature is that like hurricanes, and we're very much in in an area that gets a lot of hurricanes here, are increasing in frequency and severity as anthropogenic climate change progresses. But for something like the crystal skipper, we're not really working with a lot of room. We don't have exactly a lot of wiggle room when you're talking about the species whose entire range is 30 miles of barrier island. So if those like increasingly severe storms come in and dramatically impact survivorship amongst like the caterpillars, then we're not exactly dealing with a lot of room to work with. Like one hurricane could easily cover its entire range, and so those are things that really compound on each other for something like this. And I think it's always important to note that the biosphere isn't dying, it's being killed, and that's something that we should do something about. And then as individuals, creating that culture of caring, and a connection like with your local ecology is always huge. People are always asking us what we do, which is very understandable. There's all these signs saying, like don't walk in the dunes, don't walk in the dunes. And then we come out, with our highlighter yellow vests, all sweaty, carrying all sorts of contraptions. And of course, people want to know what we're doing. So, we end up having, frequent conversations with members of the public. And I can almost, write the script. There's a butterfly only found in this area. And the second you tell people that, and then just engage them with more information, it really gives people that sense of connection and like admiration, like of the uniqueness of the area in which they live. And when you have that culture of connection, people tend to take conservation very seriously, especially what if they've got young kids there and they're like, well, I love butterflies. There's a whole like cascade of caring that can really happen there. And then for people who do live in that area, you can certainly manage your backyard for wildlife. Again, a big part of this project is, we want to have the data to tell people, like, oh, plant more of the host plant, plant more of these flowers. So, we're certainly working on, how we can do that for this specific species, but, no matter where you are, there are things you can do to manage your backyard for wildlife. And then similarly, citizen science is always a great thing, like the Great Southeast Pollinator Census, like another, shameless plug there, for this. So there's certainly things that we can do. We don't want to feel helpless. And so for myself and the rest of the crew, thank you very much. So I want to, shout out, obviously, Laura, this is her PhD project. And without, the enormous amount of effort she puts in, none of this would be possible. As well as Dr. Price and Dr. Youngsteadt, who are, always out there in the trenches with us whippersnappers, showing us up. So yeah, thank you from all of us. And then thank you as well to our partners shown here.
That is so awesome, Doug. And I can't thank you so much again, all of our speakers who have joined us today, and for everyone who's joined us, you've gotten quite the spectrum of of topics, all the way from like looking at it at landscape level all the way down to a few dudes in North Carolina. And I hope that I actually had read Doug did a really nice write-up in one of the local newspapers out on the coast, and that's how I found about it. And it really, you made a really great point, is when people know that they have they get at the sense of ownership of, this is in my backyard, this is in my community, and this what makes my community unique. I think for those of y'all who have that seems to be like a reoccurring story that people share with me as they participate in the census, is they start noticing the things in their community that they may not have ever noticed before, or they notice, they don't see as much pollinators as they used to, or they see more than they used to see, and they don't know if they've just always missed them, or so it really can be empowering to just take a moment. And we appreciate all of your work, and ecologists like yourselves. And for those of y'all who have young people in your lives, we still need more ecologists being trained. So ecology is a viable career choice, and we need more young people in this line of work. And I think these folks are demonstrating that. So before I like totally forget to go through the Q&A and and everything, Charlotte, do you want to pop on and walk us through some of the questions that we had while folks were talking?
Definitely. So they've all been answered, which is the great thing. But when we started off and you were talking about the Great Southeast Pollinator Census, there were a few questions about how to participate there, including things like, how do we avoid counting the same insect over and over? And actually, you don't. That is part of the way the census works. So you count every time an insect lands. Becky answered, that question. So I believe everybody will be able to view the answered questions in the Q&A. But so that was one that I thought was worth pointing out. So you count every time an insect lands, even if it's the same insect landing over and over.
Mm-hmm.
And there was also a question again about, do you look at the whole plant, or do you focus in on an area? And I know in the past, Amanda, you have recommended thinking about a beach ball and an area the diameter of a beach ball.
Yeah.
So you don't have to try to look at a huge group of plants, just focus on an area that's realistic for you to count.
Mm-hmm.
So those were some of the questions. Then there were some questions. Hannah's done a great job responding to questions that came in about her talk. And there were a lot of questions again around, well, if I don't want to see those stems in my yard, can I take them and collect them and put them somewhere else? And so, she shared advice that, they didn't do research on what happens if you take the stems away, but the recommendation is to put them somewhere they sit upright and stay dry, because if you lay them on the ground, they're much more likely to rot. I don't know, Hannah, you're welcome to jump in and add additional things, but by far the best thing to do is to leave them standing in the garden. And I'll tell you, I went from being somebody who cut everything back really low, and then participating in that research really changed, and I started cutting things back to 12 and 18 inches. And at first it did look odd, but it only took a season. And now that's what looks normal to me. So I think you get used very quickly to that just being aesthetically normal and not an issue to see it, and especially when it's paired with the knowledge that, oh, I could be providing habitat for pollinators.
Mm-hmm.
And let's see, I know, Hannah, you had responded to quite a few questions. Was there anything else you think is worth mentioning, with calling out?
Yeah, I can recap really quick because I did move through the end a little quickly. So just to recap based on the questions, we're recommending that you wait for a stem to grow, flower, and have the flowers be dead. You then deadhead 12 to 18 inches about on that stem. That's the stem that we're saying only trim in your first winter, after first frost, before last frost. And then once you trim a stem, don't touch it again. Any new growth that has new flowers the next year, you keep doing that same trimming technique. If you trim to the ground, you're not damaging stem nests because you're removing the stems before there can be nests in them, but you're also not leaving habitat for stem nesters to build nests. If you don't trim at all, you're also not damaging stem nests because the bees can't get into the stems. So if you trim, or if a stem is damaged, you should assume there could be a nest in there, and that's when you only trim once in the first winter. And then, like Charlotte said, if you trim and remove, just consider that mold is a really big problem for these stems, and predators, they can get in and then eat the larvae. So we want to keep the stems safe if you're removing them. I think those are the big points.
Yeah.
Yeah. I one of the things that has kind of popped up a couple of times is maybe more of a comment about kind of the frequency of insects. Some folks have noted that they've seen less butterflies this year in their gardens, and I know that's kind of a complicated question, but how do you respond to someone who makes that anecdotal observation? And it's kind of a question to all of you, because I'm sure there's some different perspectives there.
I mean, I can maybe just say something real quick, which is that, it's, yeah, I mean, a lot of times it's anecdotal, I'm seeing fewer of this, fewer of that, but it maps onto the data, like there are, we're definitely seeing a decline in insects. And one of the things I'll often hear folks say is like, oh, I don't see like as many like insects like hit my windshield and things anymore. It's just like, yeah, we're dealing with a real problem. And yeah, no, Lepidoptera is definitely across the board, mostly across the board, experiencing a lot of decline.
Yeah.
I'll just chime in quickly on data and trends. Like, one of the hardest things with insects is their populations fluctuate naturally so much. If you're monitoring like a pride of lions or like populations of lions in Africa or something, you're not going to see the population double one year and then halve the next year. Realistically, you'll be able to much more easily see the trend. Insects, just because was there a frost in this one particular week when the insects were emerging? Was it a particularly dry summer? Whatever. The populations are used to fluctuating massively, which makes it really important that we get good data across many years, because if you only look across 2 years and the population halved from one year to the next, that doesn't actually tell you if that is a long-term trend. We obviously have really good data now for a bunch of places to show that a decline is happening, and it's very significant. But that adds the importance of the census and continuing it across many years, because 1 year there may just be far fewer butterflies, but you have to look across maybe 10 years to see if that is actually showing a kind of a significant trend, or if it is just they're going to bounce back next year.
I'll add one last point, but I think both speakers, yeah, you already said most of what I would say. But I do want people to keep in mind that with these insects, even though we're thinking about our little gardens, it is a landscape level. And so we don't know everything that's happening beyond your garden. Maybe one year there's better floral resources next door than your garden, or maybe something happened to a host plant. Different bees, some years they just don't come out in the numbers that they do, and the next year they come out in normal numbers. So it's bigger than you. And yeah, just back to what Nick just said, of tracking things over time is the only way we're really going to know the answer. And we don't have the data that we need to answer that right now.
Somebody asked earlier on, and I didn't even see it, Charlotte, before, but the very first question that was asked is someone asked, analyzing the data that you have, can you talk about the trends that you observed over the years, speaking specifically to the Great Southeast Pollinator Census? And we really, Becky chimed in before she had to leave, and she says, usually researchers don't look for trends until about year 10. And for Georgia, this is only year 7 for the census. And in North Carolina, we're only in year 3. So we still have a long way to go. And the only way we can really speak to this is to get started, right? So we're really only getting started, and maybe this kind of piggybacks on something you kind of just noticed, you said, Nick, is we are still learning how to study this and keep this data and make sure that it's telling the same story over a long period of time. So we're still learning how to create the tools to track the information that we know we need to learn about. And that, I think that's one of the reasons why I think I've gotten so involved with bringing the census here and trying to support people, is it's one simple thing that all of us can do collectively through time, that we can really start to show these trends, because we really don't have that data like that anywhere. Okay, Charlotte, we've had some more questions rolling in, and did you catch
Yeah,
somebody
I
asked
had.
Doug?
Oh, well, somebody was asking, I was just trying to type an answer right now about the packet you mentioned.
Hmm.
And did everybody get it? And I was just saying, unfortunately not. It was the people who were selected, who had registered by a certain time, and then there was a drawing.
Mhm.
But the packet, all those materials, as you mentioned, are available for
Yes.
people to either purchase or download and make themselves.
I think, I don't know if I have actually published what was in all of that on our
Okay.
website. I'm going to make a note right now to make sure to get that up on the North Carolina webpage. There was a question that somebody had put in the chat, and it said, for Doug, is there local government or state government support for protecting crystal skippers?
Yeah, thank you. That's a great question. So right now we don't have any federal or state agency. So the Fish and Wildlife Service, will have like information on this species, but we don't have the data yet to like published to like state like this is like critically endangered versus vulnerable, all those things. So yeah, very much the goal is to like make that, kind of existing plan that Dr. Price created, like more data driven, like answer, more of those pertinent questions so that we can then make proper recommendations to state and federal agencies, such as the Fish and Wildlife Service or Wildlife Resources Commission.
Thank you so much. Yeah. Charlotte, what other questions have we gotten?
Quick question about the census. Can you do different plants on different days? And the answer there is yes. You could even do different plants on the same day, but not different plants at the same time. So each 15-minute time frame that you're filling out the sheet, you need to be focused on one species of plant. So you can do the census multiple times over both days if you want to. And we'd love to see that. But each time you fill that sheet out, just focus on one species of plant.
Definitely. And try, I mean, it would be cool to maybe do the same plant over the course of the 2 days, because pollinator visitation changes from the morning to the afternoon, and depending on the weather. I have anecdotally, actually not anecdotally, I do have data on this because I did the Great Southeast Pollinator Census data sheet on it. We have an oxeye daisy in our garden here in Lee County, and the green sweat bees only were there at the 10 o'clock in the morning census. When I did it at 2, it was all bumbles and butterflies, but it was small bees all morning. So same plant, flowering, same spot, everything, but the time of day completely changed the visitation. So you can see those stories just by doing that. Nick, this is a question and and Hannah, this is a question that popped up in the chat again too. It says, how to avoid being stung while working in your flower gardens where there are bees and wasps and yellow jackets underground nesting, but you're still preserving the nesting at the same time. What do you say to that? How can people
I mean, I've very rarely been stung out in the yard. I mean, obviously the first thing I guess is try and work. Yeah, if you know that you're allergic, then I guess, you know, there is a higher level of risk working out in your yard. Obviously, keeping clear of any nests, looking out for, you know, things like bumblebee or honeybee nests, because they're often more aggressive. But generally, I find most pollinators are really not aggressive. You can even, I mean, I go around picking up solitary bees without even worrying about they sting me, and every now and again they do, but I'm not allergic, so it's okay. Yeah, I would say for bees, it doesn't feel like a big risk for me at all, unless you're working close to a hive. Yellow jackets obviously can be very frustrating, but I'm one of those people that just keeps still and lets them do their thing.
Yeah, I'll add learning more about what to expect from their behavior can be helpful. So if they're out in your garden, they have a job to do. If they're wasps, they're hunting. If they're bees, they're collecting pollen and nectar. And they more or less don't care that you're just standing nearby. If you're messing with them, that's a different thing. And the social species, so honeybees, bumblebees, social wasps, they do have large nests that they would collect. So just be aware of your surroundings. If you know that there's a ground nest, maybe don't go there when that nest is active. But for the most part, those species, other than honeybees, bumblebees and social wasps, they're on an annual life cycle here. So the nests will go away in the winter, and then you could kind of take care of that area when those nests aren't active. So hopefully they don't use it again next year. But just observing, learning their behaviors, understanding how to predict their behavior can be helpful. And then the last thing I'll say is, because someone talked about allergies, for the most part, this isn't everyone, but for the most part, if you're allergic to a species, you might not be allergic to all species. Their venoms have different proteins, and so your bodies react differently to honeybee stings versus bumblebees versus wasps. So again, it's more just let's learn and observe, and not until we know more about the behavior and how we all respond and what's in your garden. Don't be too nervous. It's more of like a learning experience. Hopefully that helps.
Mm-hmm.
And just, I think, quickly on yellow jackets, just to plug them, because I know how frustrating they are when you're having like a picnic or something. But I don't have any data to hand, but the quantity, the mass of caterpillars and herbivorous insects that a single yellow jacket nest kind of like takes out of your yard each year is just incredible. So the pest control service they're doing, helping to keep everything in like a decent balance, is so important. So it's just a plug to say that everything out there is carrying out some kind of service that adds to the whole sort of ecosystem of your yard. So where possible, giving them some slack. But I know that can be frustrating sometimes.
And I see there's a question that's come in. I think this one's probably for Doug, that asks about, does it hurt butterflies when you tag their wings, and how is it done?
Yeah, for sure.
Man.
Yeah. So the answer is it can, particularly if you're inexperienced. So I obviously I would not recommend, we're doing this, it's a calculated risk, but we need the data, we need the information. So, and also, like we're doing this, like as professionals. So don't try this at home, kids. But it is, it's definitely, we need the information. There is like it, especially, as we're training people up, sometimes like that can, that can certainly happen. But my usual like protocol is like I'll write on their wing, and then like I'll let them like perch on my finger, and if they like take off right away, like which is the case, I want to be clear, is the case like the vast majority of the time, they're usually like very I should have brought my butterfly forceps, sorry to show people, but they're usually so happy to like get out of there that they're just they're head right off somewhere to go eat or something. But yeah, like there have been like like sometimes they won't seem to be doing like quite as well after the fed, but like then we'll basically just like set them on the host plant, and then usually after like a couple of minutes they'll recover and just fly away.
Yeah, catching insects is its own kind of skill set, and I guess this is a great opportunity. So we do have a lot of events across the state, but Hannah Levenson will be joining the North Carolina Pollinator Conservation Alliance on August 22nd at the State Farmers Market. We are doing the North Carolina Pollinator Field Day. And so Hannah will be actually leading a insect catching observation booth. So if you wanted to learn how to safely catch insects and do some ID, Hannah will be live in person at the State Farmers Market on Friday, August 22nd from 10 to 1. Shameless plug, right?
And I have a quick question, Amanda, for you. You mentioned oregano as being the plant that is collaboration this year between the Great Southeast Pollinator Census and the UK Pollinator Monitoring Scheme. So if people don't have oregano this year, if they planted it, do you think this will be just a this year plant, or do you think next year and in future years that collaboration will continue so they could plant some types of oregano to observe in future years?
Yeah, that's a great question. We don't know. I think it's going to be something that we continue every year because for the UK Pollinator Monitoring Scheme, they're pretty locked into their list of monitoring plants because they're also coming up on their they're on their 7-year mark as well, and so they want to make sure to keep that list pretty stable. And so I'm pretty confident that this will be something that we do year after year. So yeah, please, if you don't have any oregano, plant some this year. And it is a very aggressive plant. It's not quite as aggressive as mint. It is in the mint family, but it does spread. So be warned. I have seen it go from like a little 4-inch pot to like 2 feet in like a season if it really likes where it's at. So it's a great container plant. If you want to keep it contained. I have found too, I've had to trim mine back midsummer because they will sometimes bloom in June and then they'll kind of fizzle out. So if you find, like, if you are growing any kind of mint or oregano and you want to monitor it in August, probably keep an eye on it about June, July. And once it starts looking a little tired, just chop it back and it will reflush and reflower in time for you to monitor it during the census. I know we're coming up on 3 o'clock. We do have a few more questions. Can y'all join us for a few more minutes? And for folks who do have to pop off, we are still recording this, so and the recording will be sent out afterwards. So if you need to pop off, we appreciate you being here. Otherwise, we're gonna finish answering these questions that have come in. Somebody asked a really good question. This person said, I have mosquitoes in my yard. When I'm out doing the survey, if I'm using a repellent, will that affect the insects that come near my plant? It's a great question. Does anybody want to chime in on that one?
I'll say that mosquito repellent is designed to work for mosquitoes, but a lot of these products that are designed for insects, pollinators are also insects. So if something works for an insect, it could work for all insects. So depending on what you're using, and they are a repellent. So if they were impacting things that could be visiting the flowers, it would repel them from visiting that exact flower. So when I do research, I tend to not use any sort of bug spray. You just wear long pants, tuck your pants into some tall socks, wear a long sleeve shirt. So keep in mind the pros and cons of all these things.
Yeah, I'll just add, sorry, sorry, go.
Top tips from the field.
I was just gonna add, there's so many different, like, I completely agree with that. There's so many different smells we wear. Like, we don't know what effect perfume has, some that are floral scented. I found if I'm wearing certain colors of field clothes, particularly like blues and purples, I get bees trying to visit me, like landing on my shirt. So yeah, I would say, like, there's so many different ways we could affect it if we get very close to the flower, but bug spray seems like one of the most likely to do it. But I would say, if possible, not being very strongly scented in any particular way is probably helpful. But yeah, long pants is always a good thing.
Yeah.
I'll say real quick that in like all my years of field work, like especially with the crystal skipper, like I wear, again, to reiterate, like I wear long pants, like long shirt, like sun hoodie, and like I don't use bug spray, and I'm fine.
Yeah. Yeah. No, great top tips. I know that's not in the middle of August in the full blazing sun when you're sitting there for 15 minutes observing insects. If that's not something you're used to, that can be really tough. I have found that, you know, I use an umbrella sometimes when I'm doing the census, and you can usually stand far enough away and have the umbrella kind of angled enough that it doesn't cast a shadow on the flower you're observing, and I've not seen any impact from that. So wearing a big sun hat, using an umbrella, you know, just keeping the sun off of you can make a huge difference. And then yeah, just cover up and use, you know, loose fitting, light colored clothing, and that can make a huge difference. We got 2 more questions. Charlotte, what we got going on?
We got are ticks pollinators?
Oh God.
Does anybody have ever seen any evidence of ticks being pollinators?
I have not. I'll jump in really quick. To be a pollinator, something needs to be visiting the flower and then interacting where it would get pollen on its body and then go to another flower. So the other insects are things that will be on the flowers. So the general expectation of what you might see in your garden are like beetles. There'll probably be lots of different kinds of beetles. That's the big one, I would say. Spiders will be on there too. Maybe they could be incidental pollinators, but things that go to flowers.
Yeah.
Yeah. And then there's a question, will pollinator plants near a hummingbird feeder attract more bees than normal? I'm not sure if I'm wondering if their concern is, are the plants going to attract more bees to the hummingbird feeder? But just in general, I guess, if a pollinator plant is near a hummingbird feeder, would that attract more bees than normal to that area?
I can't say I know that for sure. If anyone else does, you could jump in, but those are different types of food sources and they have different structures. So I would say that the hummingbird feeder is more designed towards the feeding structure of a hummingbird rather than a bee. But I saw that Nick might have something to say as well.
Not really. I mean, I've actually never observed any bees on my hummingbird feeder out here. I think, yeah, it doesn't necessarily work with their like mouth parts in the same way. Also, generally, if you're just putting in sugar water, it's not scented. So hummingbirds are often attracted by the color, like bright red or something. It's not necessarily going to be drawing bees away from your flower. So if you were counting hummingbirds, then yeah, the hummingbird feeder would obviously mess with your data, but I can't see any reason why it would impact insects significantly.
Yeah. And that's a good point. Kind of nice there's nothing else, but the note of the hummingbirds, one of the big pieces of feedback I've gotten the last 2 years of censuses here is people ask why we don't include hummingbirds on the data sheet, and that's because it's for insects and insects only. And while hummingbirds are phenomenal pollinators, especially where Nick's at on the West Coast, and our ruby throats are excellent hummingbirds over here, that's not really the purpose of the census itself. It's to study insect populations. Charlotte, were there any other questions that we missed or wanted?
I'm scrolling back through the chat now and I'm not seeing anything new. We've definitely got everything. Well, we've got just a couple more things have come into the Q&A, but I think they're more observations than actual questions. So I think we've got it.
Okay. Well, before I finish up with a few more things, panelists, I'd like to give you, the floor one more time. Nick, Hannah, Doug, I'll let you guys pop in that order, but are there any kind of final thoughts you'd like for folks who joined us today to kind of take home?
I guess the final thing I'd share is obviously I'm in California right now. I'm hearing about North Carolina. I've spent a lot of time in the UK. Having these local census schemes can be a fantastic way of, when global problems feel like far too big for any of us to be involved with in any way. We look at biodiversity decline and all the kind of environmental and political stories happening across the entire world. We can feel very powerless to make any kind of difference. But I think these local schemes that add up to, in aggregate, have like a massive impact across the entire world are so important. And looking at like the impact of our own garden can have, our own yard, you can put a plant in and see the bees coming to it and be like, well, I've just increased their food supply. And it's like that thing that people always say of think global and act local. That's kind of the only way that we can really address environmental challenges for 99.9% of the population. So I'm so happy to hear this scheme exists, and it's similar in some ways to some things in other parts of the world, and hopefully it just gets bigger and bigger.
Excellent. Thank you.
I love that, Nick. I will say, don't be intimidated and just get out there. Don't let perfection be the enemy of good, you know, that old saying. Start where you can, and we can always do more. If you don't have a pollinator garden now, adding any amount of flowers, any plant that flowers and provides resources for bees is better than nothing, and you can always build up. I saw a couple comments where people were like, oh, I didn't know this, and I'm thinking about the things I did when I didn't know this information, and maybe we harmed some pollinators. We're all learning. I didn't know this before I did the stem nesting project either, so just start where you are. We're always going to keep learning. We're always going to keep improving. The census data, you know, is one kind of data. If you've never collected data before, just get out there and try it. And then if you like it, you can get more and more involved in other activities. So just start and then see where you go from there, but don't be intimidated.
Yeah. Thank you. Yeah.
Yeah, one final thought I might leave people with is, you know, like endemic species are often like a really cool way of like getting people like really interested, you know, in like their local ecology. And I think people just, you know, I hope that this talk like, you know, obviously shed light on one, but people often think, oh, I have to go off to some, you know, far off island, you know, of like the Seychelles, the Galapagos, you know, to see unique species. That's not true at all. So I obviously, you know, this is predominantly a North Carolina audience. We have a lot of really, really amazing endemic species. You know, from the magnificent ram's horn, only found in the Cape Fear River, the Neuse River water dog. There's a lot of real, and then you know, the St. Francis satyr, which is the other endemic butterfly species we have. Those are just a few, and they're often spread kind of, you know, and they're in different parts of the state. You know, for example, I grew up in Apex, North Carolina, which is in the Neuse River, you know, like watershed. And the Neuse River water dog is only found in the Neuse and the Tar rivers. So don't think, oh, it's just that one spot on the coast, you know, that has, you know, this interesting endemic species that can be a talking point as I try to, you know, garner interest with people. You know, wherever you are in the state, there's something really cool and worth discussing about your local ecology.
Excellent. Yeah, don't take your local ecology for granted. You know, the big specials on National Geographic and BBC Earth, you know, take you to these wild and incredible places, sure, but just think the people in those wild, incredible places have maybe never been to your backyard and would be just as amazed at seeing the Neuse and the Tar River as you would be seeing the Amazon. So yeah, definitely don't take your own backyard for granted, even though you see it every day. So thank you again, panelists, for joining us. I got a few more final thoughts. So this slide here is like your like quick grab and go. If you haven't taken anything from this, take this slide home, and it is available on the main North Carolina website that you can download and throw into a presentation that you may be offering locally. And 3 things that you can do to help pollinators with the census. You can obviously participate next week, and hopefully everyone who's been here today, if you count, it will count, which sounds so hokey, but it's true. And encourage others to participate in your local community, you know, in your friend group and your family. You know, if you're here in North Carolina and you've got, you know, family down in Georgia or Florida, like tell them to count too. Remember, Alabama has never counted before, so it all helps. And if you've been so inspired and you want to host an educational event on or before the census date, there are tons of resources on the North Carolina Great Southeast Pollinator Census website where folks have contributed ideas and activities and resources, and we're always adding to that library. People are donating stuff every day. I wanted to throw this chart up because these are the folks who, if everybody who registered from all these places counts, we would add a few more counties that have never counted before. But folks, if you're in a county or next to a county that is gray, go to your neighboring county and encourage somebody in that county to count. Let's get all 100 counties counting. So we need everybody to take a moment. here's 3 citizen science projects, if this community science projects that I promote. If you've been so inspired and maybe you can't count next week, but do you still want to support a community science project? The groundnestingbees.org. This is studying ground nesting bee populations across North America. This is based out of Cornell University, and it can be something as simple as snapping a shot on iNaturalist, but also they're asking for folks to do surveys. So if you were inspired by Doug's excellent ecology talk, they have all the guides on how to do some of the ecological surveying for ground nesting bees on their website. We've mentioned iNaturalist a lot. This is a free to sign up biodiversity organization, and highly recommend if you are out there snapping photos of the natural world, this is where you can put your photos and have them contribute to citizen community science, and scientists use this. And there's some really great research out there. They just put up on their Instagram that like a couple days ago, there's a whole new species described because somebody threw up a photo that nobody had ever seen before. So just going back to say, you just never know, what you see every day may be completely novel and totally new to science. This is another one that we're doing in our backyard here in North Carolina. It's also happening in South Carolina, Tennessee, and Georgia. And Becky has done some work with this. If you go to the Southeast Bumblebee Atlas, this is a great, like if you want to dig into that ecological studies and do transects and collect bees, they have some great training resources on there. We have local collaborators here in North Carolina that you can email and work with and learn how to catch bees safely and help contribute to this really important data set to understand what our bumblebees are doing in the southeastern United States. So all of this to say, we got a lot of work to do, but every little bit counts. So what can you do in your own backyard to help pollinators? So without further ado, I think that's all the time we have today, but I appreciate everyone being here. And if anybody has any questions or any concerns, please feel free to email me. I'm going to be answering a flurry of emails, I know, over the next couple of weeks about the census, but again, thank you so much to our panelists, to Charlotte for being here and keeping us all straight. And I hope everyone has a wonderful afternoon. Thank you so much.
And thank you, Amanda, for your leadership of the census here in North Carolina. We appreciate all you're doing to make it easy for everybody to participate, and encouraging people to participate, and providing this great way for folks all across North Carolina to get involved.
Most definitely. This is a really great project, and it's really great people. And I look forward to hearing all of your wonderful stories from your census events and your census counts in your own communities. Just keep them coming and do what you can do, and have a wonderful afternoon, everybody.
Thanks so much, everybody who
Thanks.
didn't
Thank you.
listen.